Okay, thanks so much, Dana. And I want to thank Dana and Susan Sang and the rest of GBATnet for inviting me to speak. I'm Amy Russell, as Dana said. I use she, her pronouns, and I'm a professor of biology at Grand Valley State University. I've been working in conservation genetics. Let me get my laser pointer out of here. Conservation genetics and population genetics and evolutionary genetics of bats for quite a few years, I think like 25 years or something like that. So I want to talk about some of the conservation genetic work and how you can kind of productively think about conservation genetics and some questions that you might have about using it. But what I'm going to focus on is just kind of one small part of conservation genetics, trying to address how we estimate population size using genetic data. So trying to address this question of how many bats are there in a population. Now, like I said, I've been working in bat population genetics and conservation genetics for a number of years. So it felt kind of weird to give a little bit of my own history, which is, you know, my educational history is getting a bit dated now, but I wanted to take a little bit of an opportunity to kind of brag about some of the students that I've been able to work with. So why is this not advancing? Why is that not advancing? Oh, there we go. Okay. Yay. I'm a good teacher, right? Okay, so some of the students that I've been working with over the last couple of years are shown here, some of my grad students, and they've been working on some really exciting projects. So people like Devin Jones, for example, here. Devin just recently published a paper looking at algal toxins in Myotis lucificus, little brown bats, and using chemical methods and genetic methods to look at what bats are eating to try and assess how they're ingesting these algal toxins and how they're partitioning the algal toxins in their body. Some really cool work that Devin published. Another grad student of mine, Olin Martin, just recently published her master's work looking at population genetics and population structure of pipistrelle bats, eastern pipistrelle bats, or tricolored bats. So look for that paper. I think that was Acta Chiropterologica where we published that. And then in the middle here, Susan Munster, who, as you can see, was a STAR student. She worked on conservation genetics using completely simulated data sets to really try and dig deep into some of the power of methods that people use, especially when we're looking at recent population decline with limited data sets. And so I'm going to talk a little bit later about some more of Susan's work, but she was a fantastic student. So for the outline for my talk, I'm going to focus on kind of three basic sections. First, I want to address why we might want to use conservation, why we might want to use genetics in conservation. What can genetic data help tell us that maybe field data has trouble addressing? And then I want to dig a little bit into theory. I hope it's not too off-putting, but I want to talk about these concepts of census population size versus effective population size, or NC versus NE respectively, and how those concepts are different and why it's really important to understand that difference. And then I'm going to spend the bulk of the time talking about how we estimate effective population size, different types of data we can use, and different types of methods we can use. So, there we go. So why might we want to use genetics in conservation? Well, if we look at the goal of kind of single species conservation, it's really to preserve sustainable populations. And to do that, we might need to know, or we definitely do need to know, some basic biology of the species that we're focusing on, right? Some basic questions like how many individuals are actually in a population? Is that population size changing over time, right? And population size change might be due to kind of natural effects. You see that here with this natural example of a predator-prey system. You can see the population size fluctuating through time here in both the prey species, the snowshoe hare, and the predator species, the Canadian lynx, right? But in other cases, population size might be changing over time due to maybe more anthropogenic effects, like the introduction of a disease to a new ecosystem, or the erection of wind turbines in places where bats are flying, right? So we want to understand how those populations are changing. We also need to understand what the geographical limits of a population are. So how do things like rivers or mountain ranges, these kind of landscape features, how do they delimit populations and distinguish one population from another? And genetic data can help us to answer these questions. In many cases where we're maybe looking at a species that's hard to assess in other ways on the ground, right? So like bats, for example, bats are difficult to count in many cases for a number of reasons, right? So bats are active at night. It's hard for us as visually oriented animals to count things in the dark, right? And there's ways to get around that, but in many cases we might be looking, or in some cases we might be looking at roosts of bats where the circumstances kind of concentrate individuals into small areas. So like in a cave roost, for example, in some cases we can just get overwhelming numbers of bats in a cave, right? I'm thinking of like Bracken Cave. People have attempted to count how many bats are in Bracken Cave in Texas for, over decades. There's been many different counts attempted, and those counts have ranged, those results have ranged anywhere from like 6 million individuals to more than 20 million individuals. And that's a huge range, right? Some of the difference in those estimates might be due to actual population fluctuations over time. Like I said, these efforts have spanned multiple decades, but a good portion of the difference between those estimates might be due to just the amount of error in those estimates, right? How do you actually physically distinguish 6 million things from 20 million things? At some point, it just becomes overwhelming, right? One of the good things about a situation like Bracken Cave, though, is it's very obvious where those bats are. When you have a huge number of bats concentrated in a cave, we know where those bats are. In other cases, it may be really difficult to locate bats. They may be roosting in like rock crevices and maybe just a handful of bats in one place or another. And so it's just really difficult to know where the bats are on the landscape. And if we can't find the bats, then we can't count them, right? Bats are also very mobile, and often the bats are going to be flying at really high altitudes. So again, it's hard to locate them, it's hard to track them. And even if we can get counts from bat colonies today, in many cases, we don't have any idea of what those population sizes were back in history. And so putting that current estimate in a historical context can sometimes just be impossible. So genetics can help us to circumvent some of those problems, right? And so when we think about genetics, we often, initially start to think about the inheritance of genes and traits in families, right? So if we look at these little cartoon butterfly families, we have 2 parent butterflies here giving rise to 3 offspring, and these offspring share genetic traits that they inherited with their parents and with each other from those parents, right? In another family, we see 2 parents that have just 1 offspring, but notice that this offspring, some mutation happened, right? And so the offspring has black spots on its wings that the parents didn't have. And if it's easy enough to think about these families and the inheritance of traits in these families, but these families are existing and over time intermingling in the context of a whole population, which is what we see here, right? And so over time, we see these families intermingling, we see mutations being passed on and to some extent propagating through the population. And population genetic theory tells us how population size influences genetic measurements that we might make in this population. So if we look at just say the amount of genetic diversity, how many alleles or how many different variants are present in the population at some time, population genetic theory tells us how much we might expect. And it tells us that, like, just basically, if you have a small population, you're going to have a low amount of genetic diversity. If you have a large population, then you have more alleles present, and you can kind of, you have the capacity to support more genetic diversity, right? And so theory, again, can tell us how population size are influencing these genetic measurements. And, you know, genetic diversity is a very simple measure that we might make for a population, but we can also look at other aspects of the genetic diversity, like shapes of genealogical trees and associations among loci. And I'll get into both of those a little bit more later on in the talk. But if we understand the theory and how population size influences the measurements of diversity, then we should be able to measure that genetic diversity, take those genetic measurements from real populations, and use that measurement to estimate population size for a real population. Okay, so that's basically the goal of what we're trying to do. So we're trying to address this question: how large are bat populations? How many bats are there, right? There's two ways that we can think about population size. And this first, census population size, or NC, is kind of what you might think of automatically, right? It's how large are bat populations, how many bats are there? That's the census size, right? So if you went to a bat roost and you see some number of bats, you're like, here's 1 bat, 2 bats, 3 bats, right? That's the census size that you're measuring, the number of adults in the population. Effective population size, or NE, here's the textbook definition of it. It's the size of an ideal population that experiences genetic drift at the same rate as the observed population. That's a mouthful, right? Let's table that definition for just a minute. I want to make the point first that census population size is generally what we want to know for conservation purposes, right? Effective population size, though, is what we can measure using genetic data. So there is this difference. However good your estimate of effective population size from genetic data is, there's always going to be this difference between what you're actually measuring and what you're trying to assess, which is the census population size. Okay, so keep that in the back of your head. But let's come back to this definition of NE. Oh. Okay, so effective population size, right? So we have, in this definition, we have, I think, 2 concepts that might be unfamiliar to you, okay? So one of them is this idea of genetic drift. And genetic drift is an evolutionary force. It causes change in allele frequencies in a population over time, right? Evolution. And it stems from just random sampling of alleles from generation to generation. So this figure here shows 5 different simulated populations experiencing genetic drift. And on the y-axis, you see allele frequencies. So all of these simulations start off with an allele frequency of 0.5. And then the lines show the tracking of that change in allele frequency over time on the x-axis, okay? So you can see that in each of these replicate populations, genetic drift is what's changing allele frequency, just this random sampling. And in every one of these, we're seeing pretty radical changes in allele frequency from generation to generation. In fact, in some of these replicate populations, like this one here, we completely fix for this allele that we're tracking. So what that means is that any other alleles in the population are completely lost, right? This other population with the triangles here, we completely lose this allele that we're tracking, right? So we can fix this allele, we can lose it, the allele frequency can stay somewhere in between. It might not change very much from the beginning to the end, but it might change radically, right? And so again, this is just a random change in allele frequency over time. How much change we see in allele frequency due to genetic drift is an inverse function of population size. So in a large population, we're going to see very little change due to genetic drift. In a very small population, we're going to see very radical change due to genetic drift. And so that relationship between drift and population size is one way that we can actually measure population size, right? Okay, so the next part of this definition that needs to be addressed is this concept of an ideal population. And so when we're talking about effective population size, we're looking at a theoretical population, which is a Wright-Fisher population. The characteristics of this are that the population is diploid, so each individual carries 2 copies of each chromosome. It's hermaphroditic and selfing. And so what that means is that everybody in the population has an equal chance of mating with anybody else, including themselves. There's an equal sex ratio. The population does not change size over time. So there's no population decline, no population growth. And there's a Poisson distribution of family size. And basically what that means is that most families have a small number of offspring, but there's a low chance that a family could have a larger number of offspring, okay? So let's put all this together, right? Basically, one way you can think about this is that if we have a real population and we estimate effective population size, let's say for this real population is 100. What that means is that that real population may actually have a census size, a number of bats in the population of like 200 or 500 or even like 75, something less than 100. But it's experiencing genetic drift at the same rate as a population like this, a Wright-Fisher population that had 100 individuals, okay? So essentially effective population size is giving us some kind of sense of how much diversity is in the population. It's giving us some sense of how evolvable the population is, how much potential for evolution there is, okay? And we might see a difference between NE and NC to the extent that any of these assumptions in the Wright-Fisher model are being violated. Okay, so let's set that aside. I want to talk about now, how do we estimate NE, right? And first, I want to talk about what type of data we're using, and then I want to talk about what kind of method we might be using to analyze those data. And let me just start off by saying that there are, the number of methods that are out there probably could take up an entire semester-long course easily, right? And what type of method you use depends to some extent on what type of data you have available to analyze, okay? Now, I don't want to talk a whole lot about how you generate those data. I think some of the earlier speakers in this series have done that really well, and you might look up David Ray's talk on genomics or Melissa Ngala's talk on microbiomes. Both of those speakers did a really great job talking about how you can generate like sequence data of various types, right? I just want to focus on kind of, let's put ourselves at the stage of having generated some data. How are we going to analyze those data? Okay, so I do want to talk about what those data might look like and what different types of data might look like. So one kind of overall category of data we might have are SNP data, right? Single nucleotide polymorphism data. And these can be generated in a number of different ways that are going to give you kind of different scales of data, right? So this made up data set that I'm showing here is something that you might see resulting from direct sequencing, right? So we have like sequences from 7 individuals or 7 chromosomes from a population. And you can see that these aligned data match up pretty well. This is generally the kind of level of variation we might see in a population. So we see some sites like this first site, everybody has a G, there's no variation in our sample. But what we're looking for are sites that we do have variation. So this second site highlighted in blue here, some individuals have an A there, some individuals have a G there. And at these different variable sites, all of which have at least one site highlighted in red, you can see that there's different levels of variation. So some of them have only one sequence that's different from everybody else. Some of them have up to 3 sequences that are different from the other 4, right? So this is what you might see from direct sequencing data, and kind of the scale that we would typically see from this is maybe 1,000 or a couple thousand base pairs from each locus, each region of the genome that's being sequenced. And a good data set from direct sequencing might have like a couple hundred loci, right? If you want to scale up, often for population genetics, to give you more power, you want to look at more loci, not necessarily more individuals, right? So to scale up in terms of loci, you might go to some kind of reduced representation SNP data, and this might be genotyping by sequencing or RAD-seq data, but basically you're looking at shorter reads from many more loci from your population, right? So your reads might be like maybe 100 base pairs long or even shorter than that. And you might be looking at maybe 1 million loci from throughout the genome, okay? And if you wanted to scale up even more than that, you could sequence whole genomes from some number of individuals, and that's going to give you all the variation in the genome. You might not need to look at so many individuals then to get a good sense of the variation in the population. Okay, so that in any case, however, whatever scale you're looking at directly variation in the DNA. Another type of DNA variation that we might be looking at is categorized as allelic data. And the most common type of this is microsatellites. And so with microsatellites, again, we're looking at the DNA sequence, right? You can see DNA sequences here, G's, A's, T's, and C's. But we're really assessing it in a different way and kind of characterizing it in a different way, right? So microsatellites are short repeats of a sequence motif. So in this case, the sequence motif is CT on the one strand, right? And the microsatellite, the variation comes from how many times we see that repeat, okay? So in this case, for allele 1, we have 8 CT repeats, CT, 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 just like you see, right? And so that's the microsatellite, okay? That's the highlighted in yellow part here. And so the microsatellite itself with 8 repeats of a dinucleotide motif would be 16 base pairs long. And if we include this entire fragment with the unique flanking region, we get a fragment that's 26 base pairs long. Okay, so this is one allele. It might differ from another allele, like we see here, allele 2 has a different number of repeats. So the overall fragment is shorter, right? This one only has 6 repeats. So the microsatellite itself will be 12 base pairs long. With that same flanking region, that would give us 22 base pairs, right? So it's 4 base pairs shorter than the 8 base pair, or than the 8 repeat allele, 2 repeats shorter, right? And allele 3, we have a slightly higher number of repeats, 9 repeats. So it's 1 repeat longer than allele 1, or 2 base pairs longer in terms of the overall fragment than allele 1. And so these microsatellites are inherited in a Mendelian fashion, right? Each individual would carry 2 alleles, and they might be the same. They'd be homozygote, or they might be different types of alleles. It'd be a heterozygote. When you have offspring, you pass on one of your alleles to your offspring. Your partner passes on the other allele and other allele to the offspring. And that offspring might be, again, a homozygote or a heterozygote, depending on what kind of alleles they inherited from their parents, right? Okay, so generally speaking, we have the SNP data and we have microsatellite data or allelic data. Okay, so those are the tools we're going to be measuring, right? That's what we're going to be measuring. So how are we measuring any from that data? Well, we can estimate a whole lot of different types of NE, right? There's a lot of different types of data, and there's a lot of different types of NE you might target. So in talking about types of NE, I want to introduce this concept of a kind of a population history diagram, okay? And so we're going to see these types of things several times in the talk. And so I just want to kind of orient everybody to what we're seeing here. I think all of my population diagrams have time running the same way. So we're looking at historical population up at the top of the diagram here, and then time is running from the past to the present as we move from the top of the figure to the bottom, okay? The width between these horizontal, or the two side lines, indicate the relative population size, right? So this ancestral population was relatively large, and it was constant in size, right? These two sides are constant width apart. Then at some point in time, this population started to collapse, started to decline exponentially in this case. And so we see this population decline until we get to the present day, where we have our current population size, or our contemporary effective population size. So, like, where you are in this figure, what kind of NE you're trying to estimate reflects like where you are in time, right? So you might be focused on estimating the contemporary NE, and you want to look at what is the effective population size now, or some methods are going to be more concerned with the evolutionary NE. What was the effective population size at some time in the past, right? And there are methods, we're not going to have time to talk about this now, but there are other methods that just look at the change in NE, right? Some of them don't even give you any quantitative estimate of NE. It just tells you, excuse me, is NE increasing? Is it decreasing? Is it staying the same? And in some cases, that might be enough of an answer, right? Excuse me. Okay. So let's focus on this first type of NE, contemporary NE. There's a lot of different methods to do this. I'm going to focus on one of the more popular ones, and I think one of the more powerful methods. And this focuses on linkage disequilibrium. It's a method that Robin Waples came up with. And basically what we're looking at is how associated are alleles at different genes. Now, if you've taken a kind of a general genetics class, you might be familiar with the concept of physical linkage, which is what I'm showing here. So we have a red gene and we have a blue gene, and this black line behind them is supposed to indicate a chromosome. And so you can see that the red gene and the blue gene are physically linked to each other. They're on the same piece of DNA, the same chromosome, right? And so the way these alleles are arranged, we have a big A allele at the red gene on this top chromosome. We have a big B allele on the top chromosome at the blue gene. And so big A and big B are physically part of the same piece of DNA, and they're going to be inherited together as a unit, right? Similarly, on the bottom chromosome, we have little a and we have little b linked together. Those are going to be inherited together as a unit. And so if these were chromosomes that were in parents, we would see in the offspring, we would tend to see more often big A and big B present together in the offspring, little a and little b present together in the offspring. We can also see this phenomenon of linkage disequilibrium among unlinked, among loci that are not physically linked to each other. So loci that are on different chromosomes altogether, okay? So here we have our red gene with the big A and the little a alleles. On a completely different chromosome, we have some green gene that has a big D and a little d alleles, okay? And particularly in small populations, we may see some statistical association between alleles at the red gene and alleles at the green gene. So for example, big A and big D may tend to be statistically associated together, to be found more often together than you would expect at random. Particularly again in small populations. The amount of association, the amount of disequilibrium in that association between alleles at different genes is going to be more profound in small populations, and we're going to lose that signal in large populations. Okay. And the method that Waples came up with is actually, it's really good. But it's really good with some limitations. So if you are looking at a small population, specifically a effective population size less than 1,000, this method has been shown to be really accurate and really precise. So this table here is from one of the kind of early papers developing this method that Robin Waples published. And so what they did is they're looking at accuracy, and they simulated data for a population that starts at 500 individuals, and took from those simulated data, took a sample of 50 individuals, and then separately took a sample of 200 individuals. They estimated NE, and they also estimated error around that estimate of NE, right? So the confidence interval, 95% confidence interval. And what they found is that with one of the more often used ways of estimating the confidence interval, this jackknife method, around 90% of the time, the confidence interval included the true known effective population size. So that's pretty accurate. That's really good, right? Now, how big is that confidence interval? Well, again, if you're looking at small populations, and here's an example from real data. This is something that Cassandra Miller Butterworth and I were working on, looking at coastal island populations of bobcats in Georgia and South Carolina. And we very successfully used this method. And we found, for example, on Cumberland Island off Georgia, we got an effective population size of 12 individuals. And notice the confidence interval here ranges only from about 9 to 15. That is a super tight confidence interval, right? And if we have a really strong chance of having that confidence interval include the true effective population size, then we have a lot of confidence in this estimate, okay? Now, what happens if you have a large population, right? So this method just completely breaks down very often if you have a large population. So this is from a paper that I published on Hawaiian hoary bats. And we estimated in a number of different ways, we estimated effective population size. So this is for one of the populations in Hawaii. We estimated effective population size using a completely different method and got an answer of about 132,000 individuals for current population size. Now, I took some data from this same population and estimated NE using this linkage disequilibrium method, and this is what I got, right? So 3,000 individuals was my point estimate, but I want you to pay more attention to the confidence interval here. We are 95% confident that the true NE is somewhere between 119 individuals and infinity, right? I'm going to go out on a limb and say I'm pretty much 100% confident that the true NE is somewhere between 119 individuals and an infinite number of individuals, right? I'm also pretty confident that that's a pretty useless measurement, okay? So notice that this is unpublished. This is just an analysis that I did. We got this result and we're like, this is garbage. Let's set that aside and never talk about it again. So you really need to approach these estimates with an air of suitable skepticism, right? And when the method is telling you that it doesn't have any idea what the actual value is, you need to listen to that message. Okay, so we've talked about contemporary NE. Let's move on to evolutionary NE. So again, evolutionary NE, we're trying to get at what is the effective population size at some time in the past, right? And a lot of these evolutionary NE methods use, they're based on a theory called the coalescent theory. And I want to explain kind of the basics of coalescent theory to you in 5 minutes. So, okay, so we have here just a schematic of a very small population. This is like 6 diploid individuals or 12 alleles, okay? And we're looking at some point in time, generation T, we'll call it. And we have these 2 yellow alleles in this population. What we want to know is what is the probability that these 2 yellow alleles in generation T came from the same parent allele in the previous generation, generation T minus 1. Okay, so there's 2 parts to this consideration. One is that one of these alleles had a parent in the previous generation, right? Well, that's completely certain, right? The probability of this first yellow allele having a parent in the previous generation is 1, right? Now, in order for these 2 alleles to have the same parent in the previous generation, now we've limited which alleles this parent needs to choose, right? Specifically, it needs to choose this parent in order for yellow 1 and yellow 2 here to have the same parent in the previous generation, right? This allele could have any of these alleles as a parent, but in order for it to have the same parent as this other yellow allele, in order to coalesce in the previous generation, it has to choose this yellow allele from generation T minus 1. And the probability of that happening is 1 over 2n, where n is your population size of individuals, right? So 6 diploid individuals. And in a small population of 6 individuals here, the probability of these 2 alleles coalescing in the previous generation is probably pretty good, right? It's 1 over 12 specifically. If you had a large population, let's say instead of 6 individuals, you have 60 diploid individuals. Now you have 120 different alleles in the population. And the probability of these 2 yellow alleles having the same parent in the previous generation is going to be much lower, right? Instead, they may coalesce in the T minus 2 generation, or T minus 3, or T minus 4, okay? And so the expected time to coalescence, how many generations do we need to go back before 2 alleles have the same ancestor? That's a function of the effective population size. So we can look at that time of coalescence and try to say something about effective population size. That's basically what we're doing with coalescent methods. And so not only can we try to estimate effective population size, but we can also start to get at this idea of how has effective population size changed over time, right? So here we're looking at another way of kind of drawing a population through time. Okay, again, we have the past at the top of this figure and the present at the bottom. Okay, these bold lines show us the population size. So this is a constant population. And so the width between these bold lines is consistent all the way through time. Okay. We have the coalescent genealogy drawn within the confines of this population. And you can see that these lineages are coalescing at pretty much a regular rate as we move backwards through time. On the other hand, if we have a changing population size through time, if we're looking at, say, a declining population, now in a declining population, the older, the further back in time we go, the bigger the population size is. So this ancestral population is pretty big. The current population is relatively small. And we can see the effect that has on our coalescence times, right? When the population is small, lineages are coalescing rapidly. And then as we move back through time, the population is getting larger and larger. And so these branch lengths are getting longer and longer. It takes longer to find a common ancestor when that population is bigger. On the other hand, if we look at an expanding population, you're looking at the same phenomenon, but just kind of flipping it on its head, right? In an expanding population, we have a relatively small ancestral population that grows to a large current effective population size. When that population is big, now and in the very recent past, it takes a long time for lineages to coalesce. And so these tip branches are really long, they're really extended. And the internal branches, when the population is relatively small, those are really quickly, really fast. So fast rate of coalescence is what I'm trying to say. Okay, so how's this method been used? Well, a great example comes from Brian Karsten's lab. Michael Sovic published this paper in 2016. And they were using coalescent methods to estimate effective population size for 3 species of North American tree bats. And these were specifically sampled in the Midwest. And so we have silver-haired bats here on the left. In the middle, we have hoary bats. And then on the right, we have red bats. And so one thing we see is that all 3 species fit a model of population growth. So that's really cool to see, right? We also have estimates of current population size shown at the bottom of the figure here for each species. So red bats have a really large, relatively large effective population size now in the millions. Hoary bats are kind of intermediate, several 100,000. And silver-haired bats have relatively small effective population sizes. We also have estimates of the time at which this population size started growing. And notice that for each of these estimates, we have 2 numbers shown. They're actually presenting the confidence interval for these parameters, okay? And that's great, that's super, right? But you might think like it is honestly very tempting to look at these values and say, oh, well, there's, 1.2 to 1.6 million eastern red bats out there. That population's doing great, right? Well, remember that what we're estimating is effective population size. Don't interpret these as census population numbers, okay? And I'm going to turn to my former student, Susan Munster's work to really illustrate how confusing NE and NC can be harmful to a population, right? So Susan did a whole bunch of simulation work. I'm just going to show one little bit of it where she was simulating a microsatellite data set for a sample size of 50 diploid individuals. She simulated 10 microsatellite loci, which was kind of a standard data set. It's, honestly, it's a small data set, but it's something that you might see in the conservation genetic literature. And we're looking at a declining population that's declining from an initial population size or an ancestral population size of 10,000 individuals. Okay, so what we see here on this graph is that ancestral population size. Notice that we have this red line here of the known ancestral population size. We know what it is because we simulated these data under these specific parameters, okay? And we are estimating that NA at different time points in the process of population decline. So 1 generation into decline, 5 generations, 10, 50, 100 generations. And in all cases, no matter how far into that population decline process we are actually able to estimate NA really well. You can see that these box and whisker plots, which show the results of the data analysis, they're really tight. They're right, on target where they should be. That's great, right? What about contemporary effective size? For these same simulations, now we have current effective population size over the same time points, right? And again, and the red line is showing what we know contemporary effective population size is at these different time points. And so this is reflecting that population decline. And so the known effective population size is stepping down as we get further and further into decline. The estimate of that parameter, though, is not responding to the population decline. So we see that there's really weak power with this method and with this size of a data set to address current conservation threats. Okay, so just to wrap up, I want to emphasize that these methods can be really powerful, but you need to be cautious in how you use them and how you interpret them, right? Keep in mind always that the conservation interest is typically in census size, not in effective population size. Often the census size is going to be greater than effective size, but as we see here from Susan's estimates, that's not always the case, right? The red line really represents the census size. The box and whisker plots is our effective population size estimate. And if we use this as a monitoring tool, right, we would be completely missing this signal of population decline. There are other ways you can use NE. So NE can be used to maybe indicate the evolutionary potential of a population, the amount of genetic diversity that's in a population. If you have a small population, you might use something like LD, that linkage disequilibrium method, as a monitoring proxy of NC. I'd have a lot more confidence in using NE that way. But you always need to consider the error in your estimates and not just pay attention to the point estimates. Okay, so to wrap up, I want to thank you for listening, and I want to advertise our working group, our NE working group, NUGE. I came up with that. That amuses me. And this is run by Francisca Almeida, Balaji Chattopadhyay, Elise Lauterbur, and myself. And if you're interested in participating, we're working on a couple different manuscripts targeted at managers for one of them, and at any users and people who are actually using those methods for another paper. We're also working on some workshops. So please reach out to me, and we'd love to have you involved in that effort. So thanks. I'm happy to take any questions.
All right, thank you so much to Amy. And we definitely have time for some questions today. So you can either feel free to unmute yourself, or you can pop some questions into the chat if you like.
There we go. Now I can reach the chat window.
This was excellent. Thank you so much, Amy.
Yeah, thank you.
And while we're waiting to see if there's...
I see people in the chat window are just being very kind, but does anybody have any questions for me?
Yeah, I do, Amy.
Yeah.
How many bats are there in the world? I want an answer to the total question.
Oh man, that's I abstain from answering that question. There's a lot of bats. Like there's some of them are not very numerous, but there are some that are just enormously numerous and having humongous ecological impacts. So yeah, it's all over the place. And having a lot of bats in a population doesn't necessarily mean that that population is safe from conservation threats, right? Just think about like passenger pigeons, to channel Gary McCracken for a minute. You can have very large populations and lose those in just a really troubling amount of time.
Maybe a follow-up question for Nancy. How many bat species are there?
Yeah, what's the current number?
What is the current number?
Well, the current number is 1,469, but I know of at least three or four more that need to be added for the next update in January. So I think we're definitely over 1,470 now.
Awesome.
That's so cool. All right. Well, If there's no more questions, I'll just go ahead and like, again, Amy, thank you so much for kind of being our last talk of the year. It was absolutely fantastic. And
Thank you.
again, a couple of those reminders, we won't be having our next GBATnet webinar until February. So stay tuned for the new year for announcements. And again, if you are in contact with or are a student or have students in your lab, please remember to spread the message about the GBATnet Student Scholar application. So those are going to be due in mid-January, and you can find all the information of that online. And if you want to come back and reference Amy's talk later, which I think I'm going to be, it is going to be uploaded onto the YouTube later on this week, and the PowerPoint will be available as a PDF online as well. So feel free to access that later on.
Thanks, everyone. Thank you so much.
Thank you everyone for being here. Have a great rest of your morning or afternoon or night. Bye, everybody. Thank you so much.